All right, so I am Mriganka. I know my name is a little bit different, I guess. But I'm an assistant professor here at Minnesota State University, Mankato. So I'm not from University of Minnesota. I'm from here, Mankato, all right? And those of you who doesn't know that, okay, here at Minnesota State University, we have an agriculture program that we started actually. If you do not know, I just want to tell you about that one, okay? And I joined here 2020 fall. So I'm pretty new. I'm learning about Minnesota. And this is a great opportunity for me to meet all of you. Finally, we are meeting in person, which is great. I know. I mean, I wish this room was full. Some of the people are attending, I guess, through Zoom, which is great. All right. So I think you all kind of learned about different topics about nutrient management and all, but I'm going to switch my gear and talk about soil health. And what I'm going to talk about that how we can manage the soil health for enhanced nitrogen cycling. But in addition to that, I'm also going to talk about some of the other ecosystem functions which could be related to either nitrogen cycling or maybe the soil health itself that can provide all the ecosystem benefits. All right. So the way my presentation is outlined, so I'm going to talk about what is soil health, followed by I'm going to discuss about what is the importance of soil health. And then I will talk about how we can increase the soil health with the help of some research examples. And most of the research that I'm going to show here, all my data, that should be based on Iowa State University. Because before I joined here at Minnesota State University, Mankato, I was a postdoctoral research associate at Iowa State University. And as I said, I joined here 2020 fall, then the COVID came, didn't do much research, but the great thing is I already got a lot of great students, and I have here 2 students who are sitting here. Can you wave your hands? All right, so they're also excited. This is their first actually conference. All right, so they are now working on some of the projects. Hopefully next year, maybe sometime soon, if there are some other conferences where I can bring my own data from my own lab here at Minnesota State University. And then finally, I'll be concluding my topic or my presentation. And if you have any question, I will try to answer all those questions. The first thing first, soil health. If you think about, that's the buzzword right now, right? It's trending everywhere, I guess. And if you think about it, it's not a new term. What I say sometimes that, okay, we are just reinventing the wheel. All the terminologies, it's been a long time. People were always talking about like soil management, soil tilt, or like soil quality. There are like so many other different names that we have been talking about. So this is the data that I'm showing you. We actually published as a book chapter with Dr. Doug Carlin and my previous supervisor, Marshall McDaniel. It was a book chapter, and where we kind of got this data that how many times these words, the soil health, the soil tilt, or the soil quality has been used. And you can go to Google. So there is a thing called Ngram Viewer, and you can actually check these different names. Sometimes I make actually fun with that, like, all right, Shakespeare, or like maybe some other, big famous guys. You can see how many times their names has been used. You can do that. So if you look at this green line, that is your soil health. That's the term. And if you see it, last few years, it is kind of exponentially, it's been increasing that term. All right. So since we are talking about that, the first thing is, what is soil health? Which is very important to know, right? So soil health is also referred as soil quality, as I said. So it is defined as the continued capacity of the soil to function as a vital living ecosystem. So if you think about it, it needs to function. And at the same time, it has to be a living ecosystem, right? So only living things can be healthy. What I say in my class, always when I teach that, I say, all right, you are a human. If you go to a doctor, when you get sick, what do? They do a lot of tests, right? They take like blood pressure, they do blood sugar, or so many other things, like maybe your temperature for fever, and then maybe some other test. And depending on that, then the doctor will decide what problem you have. And then the doctor will prescribe you some medicine. So same thing with our soils. So if your soil is a living body, if it is not like a non-living, that's what I always say, don't call it dirt. That's non-living. All right, we need to accept that okay, we are talking about soil health. It's a living body. So only living things that can be healthy. So some of the things, if we talk about when we are thinking about the soil health, if we look at that in our soil, in an ideal condition, it has either the mineral particles around 45%, it has the pore spaces. Inside the pore spaces, we have the air and we have the water. And within that pore spaces, those air and water, other than that, there are a lot of microbes they can also go and enter, who are actually playing a great role or transforming a lot of these nutrients which is present in our soil. But when we think about the soil as an ecosystem kind of, or like a microenvironment which is providing over there, it is only 5%, like in an ideal condition. But we are highly privileged that we are living in Minnesota or Iowa or any other Midwestern states. We have a very high organic matter. And when we are thinking about the organic matter, we have a lot of these like either decomposed soil organic matter, or there might be some roots or some other organisms. And when we are thinking about the organisms, we have these huge organisms or huge set of organisms which is present in our soil, whether it can be bacteria, whether it can be fungi, whether it can be actinomycetes, protists, some soil animals, they're all present. So we need to keep them appropriate food so that they're all living. All right, so soil hosts 25th or 25%, or like 1/4 or the quarter of our planet's biodiversity. It's crazy kind of, right? Which we cannot think about. Did you know that in your 1 tablespoon of soil, it contains like almost 100 million to 1 billion of bacteria? So think about if you have your farm, we need to take care of that farm. If you have like hundreds of acres of farms, we need to provide them appropriate food, appropriate diet, appropriate management practice so that they're healthy, so that they can provide you a lot of these different ecosystem benefits. So all these organisms, I tell them that, okay, they're my unsung heroes. They're constantly working. You cannot see them. They're beneath our feet, but they're providing this $1 billion of ecosystem services, which you might have seen this kind of wheel, which is given by FAO, or the Food and Agricultural Organization. But if you look at these different soil functions or all these ecosystem benefits, some of them that we are interested in, all right? So if you look at it, that the provision of food, fiber, fuel, they're providing us, they're doing a lot of this nutrient cycling, they're regulating the climate, they're also habitat for the organisms. So mostly all of these that I have highlighted here as a soil scientist or as an ecologist, as an environmentalist, we are mostly caring about those things. And I'm going to talk about when I start showing you all the data based on my researches, which I have done when I was at Iowa State University. Mostly it will be focused on some of these ecosystem functions, including the nutrient cycling, and I'll be majorly focusing on nitrogen. All right, so if you look at it, the previous slide, the wheel at the center, it was the soil organic matter. So we can think about that the living soil organic matter, that is the foundation of our biodiversity or the foundation of our health, which is very important. Because healthy soils, they are the basis for healthy food. Think about right now, our population is almost like more than 7 billion people. And we are estimating that by 2050, it might be going up like more than 10 billion people. If there are like some farmers who are sitting in this room or maybe present online, we need to thank you guys because you are taking care of us. All the population which has been increasing, you are growing the food, you are investing your blood, your sweat, everything. That's why we are here. So when we think about to feed all these huge amount of population, we need to think about that healthy soil is the key of success, or healthy soil is the key to feeding all these many people. Side by side, if you have the healthy soils, that means you are getting the healthy food. And if we are getting the healthy food, that means it is important for healthy people. We all want to be healthy, right? I mean, yearly, we all are getting old, I guess. I also go to a doctor, like healthy my body checkup, like yearly. So we want to see that, okay, what are we eating? When you go to a grocery store, when I'm buying something, I always make sure, okay, what's my vitamin in my food? What's my cholesterol level? Or what are the other minerals which is present in my food? But how are they getting that? So all the plants that we are growing in our soil, the soils, if it is healthy, that means they're providing a lot of these nutrients, and some of the nutrients that we call like macronutrients, and then the other one we call it micronutrients. Like you have learned today, I guess, from morning, a lot of these nutrients like nitrogen, phosphorus, potassium, calcium, magnesium, sulfur, they are my macronutrients. Also, we have a lot of like micronutrients like copper, iron, boron, zinc, and so on. So if you have a healthy soil, that means plants are getting all these nutrients for their growth and development. Now, if you think about it, your body, when you're eating all those foods, our body is made up of a lot of those nutrients. Like out of like 11 elements that I'm showing you here, that constitutes like almost 100% or like 99.9%. And out of that 99.9%, almost 99% is coming from that hydrogen, oxygen, carbon, and the nitrogen. And that is being provided by our soil or by our water. So we need to take care of that. I'm going to show you some of those examples that how the soil health and water quality, how they're interlinked, which is very important. And that is also one of my major research interests. But we have also a lot of challenges, right? Because population is growing. We need to feed all these people, but land is decreasing. Not only in the US, but throughout the world or globally, it's been decreasing. We also have a lot of other problems like erosion. We know in Minnesota, we know in Iowa, Midwest, every year we are losing our topsoil. It is kind of scary. So we need to take care of that one. We have a lot of other problems like salinization, compaction, acidification, or any kind of nutrient depletion which is happening. So that means we need to deal with all these challenges. And the only way that we can do it if we can take care of our soils. If we can make our soil healthy, maybe we can fight with these challenges. Now, to do that, what I'm going to show you is how we can do those things. So if you are a land manager, and if you're asking these questions to yourself, or maybe to your neighbor farmer, okay, is my land responsive to the management? And if your answer is no, what are the options? And you can go through CRP or the Conservation Reserve Program, or maybe restored grassland. All right, so I do not need to go through the CRP. I'm pretty sure everybody's aware of it. What is CRP? And when we think about the 5 principles of soil health that NRCS have given that one, if you look at it, that all the CRP, the restored grassland, everything is kind of fulfilling that one of the 5 principles of soil health, which is the continuous life plant or the root or the perenniality. So I'm going to show you some data from my research, which I did when I was at Iowa State University. So the question that we were trying to answer at that time, like, okay, how long does it take for soil nitrogen to recover from cultivation? And this project was a collaboration between Iowa State and North Dakota State University. So it was like Dr. Larry Siacek from North Dakota State University, and I was the postdoc. So we collaborated and we worked on this project. So the hard part was done by actually Larry's graduate student. And we collected all like 241 soil samples from these different parts of Minnesota, like southern part of Minnesota and northern part of Iowa. That's where we collected all the samples. And once we collected all these samples, then we analyzed them for different kind of soil health indicators. The ones that I'm going to show you right now, mostly related to nitrogen, as I mentioned before. So the first one that I'm showing you here, and next few slides, exactly the similar kind of figure that you would be seeing. So this is the inorganic nitrogen, which is kind of a snapshot of how much amount of the plant available nitrogen is present in your soil. Or another way, how much amount of nitrogen is being lost through leaching. That kind of gives you that snapshot. All right, so in this figure, your x-axis here, that's your years in CRP, or we also call it like a chronosequence study. All right. Also, within the same spots where we collected the samples, we had some topographic differences. So there are like 4 different topography that we collected our samples. One was shoulder, back slope, foot slope, and then toe slope. All right. So y-axis here is our inorganic nitrogen concentration. And when I'm saying inorganic nitrogen, that is the summation of ammonium and then the nitrate nitrogen. I think when Melissa was talking about her manure and I entered into this room, I was seeing that, okay, she was talking about the different fractions of nitrogen. It kind of made my life easy that you are already aware of it. What are the different fractions of nitrogen is present. So usually plant, when they are taking up any kind of nitrogen, they are most of the time they're taking it up as inorganic form, which means either ammonium or the nitrate form. So combining those two, I'm showing it here. So these purple or like reddish kind of color, whatever you think about it, that's your zero. We have designated it because it's the corn soybean rotation or the cropland. And as we move forward, the cropland which has been converted into CRP. We have these different years, like 2 years in CRP, 3 years, 6 years, and so on, up to 40 years. And then we had these native grassland. So what did we see? So when we did some data analysis, some statistics, so we found that years in CRP or the CRP duration, it did not show a significant effect on inorganic nitrogen because our p-value was 0.65. Right? So that means 40 years was not enough to see these changes, even though you see that, okay, they are almost achieving the native grassland. Annual decrease, we see though, that there is some annual decrease of 0.03 milligram per kilogram of soil of inorganic nitrogen pools, which has been decreased over the years. So when it is decreasing, that is actually good. That means it is suggesting that there is less amount of residual inorganic nitrogen. And which means there is like less amount of nitrogen is being lost, or it's a more tighter kind of system or tighter nitrogen cycling, which is happening within this system. Topographic position, it always had a significant main effect on our inorganic nitrogen. But overall, if we think about it, this inorganic nitrogen was not a sensitive indicator for CRP or this kind of chronosequence or chronotopo sequence kind of study when we think about it. Then we thought that, okay, we will do another kind of indicator test. That was a potentially mineralizable nitrogen, which is basically a proxy of the in situ nitrogen supplying power and is a measure of nitrogen liberated in the labile soil organic matter. And exactly the same way, as I mentioned, your x-axis, how many years they are in CRP, y-axis is your potentially mineralizable nitrogen. And when we see the data, the first thing that we found that CRP duration again didn't show some significant effect on PMN. And 40 years was not adequate time for reestablished grassland to fulfill or to fully recover. But what we saw that there is a trend that it is gradually increasing, which is a good thing, because that's the active nitrogen which might be available for the plants. All right. Topographic position, again, had a significant effect on PMN, but still this PMN was not that sensitive. So think about that 40 years. Sometimes people think that within 40 years, we will change the soil health. It doesn't happen. So that's one of the take-home messages that I want to say. Patience is needed. All right. Whereas when we look at, because that PMN, when we measured that, that was based on a lab-based incubation study. And from the same incubation, you can measure the potentially mineralizable carbon. All right. Which is an integrated measure of microbial biomass or the activity or the availability of the active carbon which is present in the soil. And in this case, we saw that, okay, CRP duration now, it showed a significant effect. It was kind of surprising to us because PMN and the PMC, they were measured exactly the same way or from the same kind of sample vials that we used. And we thought that was our hypothesis, the way the PMC would be working, PMN is supposed to be working in the same way, but we didn't see that in the previous slide. But in this case, we see that PMC was an early indicator of soil health. It was sensitive to the variation in management and annual increase of 5.47 milligram per kilogram. And we see that the changes started to happen like in between 13 to 19 years. Take home message, okay, when we are deciding some of these soil health indicators, it's really tricky. So we really need to go further to learn more about these kind of systems. All right. Now, the next thing that I'm going to talk about is another project, what we did at Iowa State. And this project was like established in around 2008 to 2010. It was called COPS project, a comparison of biofuel system, like Dr. Matt Helmer, Dr. Michael Thompson, they're part of that project. So when I went there in 2017, I joined there as a postdoc. I wrote a proposal for Iowa Nutrient Research Center because I was interested in water quality thing. Because before I went to actually Iowa State, I was doing my PhD at University of Florida, and my whole PhD was on water quality. So I was always excited about water quality thing, you know. But at that time, I was working with a septic system. And when I came here, it's all agricultural system or agro-ecosystem. That was the first time I got introduced to agro-ecosystem. So what we did in that proposal, when I wrote that, it got funded, good news, but now I need to work. So what we did, we had these different kind of system, management system, like corn, soybean, then continuous corn with cover crop, and then we had prairies. And this is the data that I'm showing you, nitrate, nitrogen concentration, which is in the tile drainage system. So all the tiled water that was coming out from these systems, I was collecting them monthly, and the data that I'm showing you, the x-axis, that is the time, y-axis is your nitrogen concentration. So what did we learn out of this thing? So we found that, okay, any kind of conservative system, that means the prairie system, where we saw that at the end of the year, the nitrate concentration was minimal. Look at it, the concentration here, we cannot even see it. It's like 0.1 milligram per liter or like 0.1 parts per million. Look at the corn soybean system, it's around 7.1. When you have the corn, continuous corn with cereal rye, the cover crop, it was around in between those two systems, so two extreme. So one system, we call it like leaky system, whereas the other one is more conservative system. But if you have the cover crops, it kind of protects that nitrate leaching. Now, the next thing that I thought, all right, so that means this much amount of nitrogen is being released through the tile drainage system. But all these tile drainage system, they are connected with the nearby streams, right? So what will happen when that end up going to the nearby streams? So there are different factors which might impact on the stream water quality. So three major factors. One, the management practice. The second one, the prior stream condition. And third, how the mixing volume or the proportion of the tile water that is getting mixed with your stream water. So what we did, we chose like three different types of stream. One is like low impacted, another one is medium impacted, and then the third one was highly impacted stream. And the way we characterize those three different categories, the low impacted stream, they had this less than 5 ppm nitrate nitrogen. The medium impacted is in between 5 to 10. And you know the EPA permissible limit for the nitrate is 10 ppm. So anything more than 10 is highly impacted stream. So we chose that. We selected those three different types of streams. We went there and then we collected our samples. And then we mixed them, the tile water and then the stream water mixed together in a bottle. And then we did an incubation study. And the mixing ratio that we had, we chose three different mixing ratio. One was 5%, one was 10%, and 25%. Or in other words, like 1:20. If your 1 part is your tile water and the 20 parts would be your stream water. All right. So that's our, it was a 14-day incubation at like 28 degrees Celsius. So we added tile water, then stream water, we mixed them, and then we separated. So that was my before incubation, or we can call it like T0 or the time 0. And the two potential kind of indicators that we chose, which can cause like your eutrophication or like the algal blooms that you see in your nearby streams. So we chose chlorophyll A and then the dissolved oxygen. And then we incubated them from 14 days in a growth chamber, 12-hour, kind of dark and light cycle. And then finally, again, after incubation, that means after 14-day time period, we measured again the chlorophyll A content and then the dissolved oxygen content. And that's how it looks like. All the mason jars, those different mixing volume, depending on the prior stream condition, depending on those different management practices, we mixed them, incubated them for 14 days. And if you look at this, I'm not sure how much it is visible. You can see some of these green patches at the bottom of the bottle. So we kind of scrape it off. We use some filter paper, and then we filtered. And whatever was storing on top of the filter paper, then we analyzed them for chlorophyll A content, all right, which is an indicator. So here is the data. So what did we learn? So here our x-axis, we have these three different types of stream. One is low impacted, medium, and then the high impacted. Within each stream, we had those different mixing volume. One was 5%, One was 10%, and then another one was 25%. And our y-axis, chlorophyll A increase. So what does that mean? So if you think about 2 time points, we calculated. One was T0, another one was T14. So T0 versus T14, how much amount of the chlorophyll A was increasing? That's what I'm showing you here, all the data. And these each color here, the darker the color, that's your no cover crop. Lighter color is cover crop. And then even the lighter color is our restored prairie. Okay, so what did we learn? First thing, the cover crop and then the restored prairie that decreased the chlorophyll A almost like 43 to 46%. Second take home message, that the low impacted stream, it was like almost 100% more sensitive to the tile water. One analogy that I always give, if somebody is trying to lose their fat from their body, right? If you have a lot of fat and if you start losing that one, if you work out, you will lose a lot. But if someone is like skinny, but still thinking like, all right, I need to be in good shape, you cannot see those changes that much. Same with here, like in Minnesota soils, our soil organic matter is pretty high, right? So these changes are pretty tricky kind of sometimes. Same thing is happening here, low impacted stream, where our nitrate concentration is less than like 5 ppm. If that gets exposed to all these different types of tile water, depending on the management practices, that means it is going to impact highly on those streams as compared to the medium or the highly impacted streams. When we look at the dissolved oxygen concentration, which is the bottom figures here, here we see the DO drop. So the first thing that we learned, that the cover crop and then the restored prairie that reduced the 14-day DO or the dissolved oxygen. Second thing, streams without the high nutrients, they are more sensitive, as I explained previously with our chlorophyll A content. It's kind of following the same way. And this 10% reverses everything. If you look at this, all these cases expected that the tile water effects. So what did we learn? That conservative or the conservation practices that reduce the eutrophication or these algal blooms. But we need to think about those 3 major factors. I mean, there might be a lot of other factors, but those 3 major factors. One is the tile water source or the management practices from where it is coming. Second, the prior stream condition. And then the third is the mixing ratio. All can regulate these indicators. All right. So that was like, okay, if your land is not responsive, so then we can think about that the conservation reserve program or the prairies, and you can see a lot of these ecosystem services, whether it can improve the soil health, it can improve your carbon sequestration, improve the water quality, some of other stuff that I have included. Maybe I didn't show the data, but it is already kind of proven that it helps in wildlife habitat or maybe climate regulation. Now, if your answer is that no, my land is not responsive to the management. Sorry, I said wrong. If your answer is yes, that it is responsive to the management, then what you can do? Can you adopt the NRCS soil health promoting practices? All right. If the answer is yes, what are the options? There are a lot of options, but what I'm going to show you based on my research, based on this composted manure and then the diverse crop rotation. So these are part of these 5 principles of soil health, where these 2 important things. One is the plant diversity as well as the livestock integration. All right. So I'm going to show you these data, how these impact or the diverse cropping rotation, how does that impact on soil health? And when I was there as a postdoc, she was the graduate student, Rebecca. So she and I, we collaborated on this project. Now she also graduated. She's now working for a company now and very successful. So this project was in a Marsden farm in Boone, Iowa, which is part of the Iowa State Farm. All right. And that study site actually was established in 2002. And if you all know Dr. Matt Liebman, he is leading that project for a long time. So what did we have? So the first treatment that we had, it was a conventional 2-year rotation, where first year we had corn, and then the second year we had soybean. But all these fields, they were kind of supplemented with a lot of these nitrogen fertilizer, and the source was urea ammonium nitrate. Based on the soil test, they were added all this nitrogenous fertilizer in these fields. Second treatment, it was the diversified rotation or the 4-year crop rotation, where we had first year was corn, second year was soybean, third year was oat, and when oat was being harvested, then alfalfa, and then the fourth year it continued with alfalfa. And it was all supplemented with majorly composted manure, like almost 85% of the nitrogen source was coming from the manure. And very small amount based on the soil health test or the soil fertility test, the rest was coming from the nitrogen fertilizer. So think about the diversified cropping rotation, and we integrated the livestock. We just published that paper. You might have seen that. It's on Twitter or maybe other forums. We have been sharing this thing. The Heart of the Press, just published like 2 weeks ago, which is a great thing. Yay. So some of the data here. The first thing that I'm going to show you is soil carbon and then the total nitrogen, because carbon and the nitrogen, they're interlinked. They're always going side by side. You cannot separate one to another. They're like husband, wife. All right. Now, so here is 0 to 15 centimeter depth, and then the 15 to 13 centimeter depth from where we actually collected the soils, and that is showing you the percent soil carbon. And that is the average of the year of 2017. And what did we learn? That compared to the conventional system, which is this, this brownish or reddish kind of bar, whereas this blue one, which is your diversified crop rotation, it showed that it increased the soil carbon almost 29%. And when we look at the total nitrogen percentage, we see something similar to the carbon. That compared to the conventional system, the diversified system increased almost 16% total nitrogen. Take home message, that means diversified system, it has the potential for increased soil carbon and nitrogen storage in the soil. Then I'm showing you here some of the data on inorganic nitrogen, because that's the snapshot of plant available nitrogen, as I talked about that before. So if you look at it, this is in 2017. That's the time when me and Rebecca, we were collecting all these soil samples, and we collected samples monthly. So the first graph, it is at depth 0 to 15 centimeter, or the root zone or the rhizosphere, and then just below that 15 to 13 centimeter. And in both cases, what we learned that compared to conventional system, the diversified system actually decreased almost 35% of the total inorganic nitrogen. So what it suggests, that means there is less residual N and fewer N losses, or in other words, diversified system integrated with livestock, that means that shows the tighter nitrogen cycling. And then this is the data that I'm showing you, which called the soil pore water, or the soil water data. So there is this lysimeter, which we install, and then we collect the water data monthly. And then we analyze them for nitrate nitrogen in that water. And all these lysimeters, they are installed like around 3 to 4 feet depth. So pretty much the subsurface drainage system where it is, or the tile drainage system. So we wanted to see how much amount of the nitrogen which is going through the soil profile. And if there is a tile drainage system, how much would be coming out from the system? All right. So we learned again, as compared to the conventional system, the diversified cropping rotation, or the diversified cropping system, that decreased around 25% nitrate nitrogen in the lysimeter water. Which is a great thing. So that means there is a reduced potential for nitrate leaching from the diversified system. So take home message, that crop diversity is very important for improved soil health. And what I'm showing you here, I'm not going to explain these things, so don't get scared. All right. So this is a big wheel, which is showing you, it is already in that paper. If anybody is interested, please go to that paper and take a look into this, that we collected almost 21 or more soil health indicators, which could be different physical, chemical, and biological soil indicators. And each and every case, pretty much we saw that the diversified crop rotation showed that, okay, it is improving the soil health. Another take-home message that I want to say here, that the policymakers or the industry, they must find the ways so that we can provide the incentives to our farmers, so that we can promote that, okay, farmers are practicing diversified system, including the livestock in their own farm. All right, so what did we get? A lot of ecosystem services out of these kind of systems. Whether it is improving your soil health, it's improving the carbon sequestration, it's improving the water quality, yield. I did not show you the data, but if you go back to the paper, actually diversified system improved the yield. Now, what if that, okay, your answer is no, that, okay, you cannot adopt NRCS soil health promoting practices. What you should do then? So there are some novel ecological approach that you can do, which I added, like some soil amendments. So one of the things that I'm going to show you here is glycerin or glycerol, which is a biodiesel byproduct, or you can think about like a liquid cover crop. So glycerin, you know, it's a sweetener. You all might have heard about it. And previously, like I think World War I, World War II, people were using glycerin for TNT or trinitrotoluene, which is an explosive. Now we have a huge biodiesel industry where we are producing a lot of biodiesel. And at the same time, there is a byproduct, which is this glycerol, which is a hydrocarbon. Or it's, in other words, simple words, is carbon-based material. Now our microbes, they're always looking for carbon for their energy. Just like us, we all human beings, we eat the food because we need the energy. Otherwise, we cannot function. That's our fuel. Same, the microbes, they need the carbon. All right? So what is the use of this crude glycerin? Well, here in Midwest, what we see, if we look at this kind of figure, that soils are most vulnerable for nitrogen leaching. And two major times. One is the late spring or maybe earlier the mid-fall. So late spring, because the snow starts to melt. At that time, any kind of nitrogen which is stored there in your soil, that is being lost. Or like early fall or the mid-fall, when crop is already being harvested, that means there is no plant is there to take up the nitrogen. Now the question is, can we manage the soil health by adding external carbon to store the nitrogen temporarily? Yes, we can do that through nitrogen immobilization. So you might have heard about the term mineralization, where any organic form of nitrogen which is being converted into inorganic form. Now, immobilization is totally opposite. That means any inorganic nitrogen which is present in the soil, we are converting into the organic form or in the microbial body. So if we can do that, we are great. That means any nitrogen which we are losing in the form of nitrate form, which is not good for our water bodies, then we are successful. So we want to see if that is going to work with this glycerol or not. So what we did a study, and it was a lab-based incubation because it was our proof of concept. There is not much study has been done. There's some work has been done in Canada and some other parts of the world, but not much has been done in the US. If I'm not wrong, maybe we are the first that we studied this one kind of. All right, so this is our proof of concept. So there are a lot of questions that you might have that I cannot answer because we haven't taken it to the field yet. I mean, we have started. Iowa State is already doing that. My previous supervisor, Marshall McDaniel, and his research group with John Sawyer, they are working on it. They have taken it to the field. All right, so coming back to our proof of concept. So what do we did? We collected two types of soil. One was the Clarion, that's the series, or the texture is loamy soil, where we had like high soil organic matter, around 6%. And then we thought we will take another soil type, which is kind of a sandy type of soil where we have less amount of soil organic matter, around 2%. And then we incubated that for 100 days. And what we did, we added different treatments of glycerol carbon as well as some nitrogen treatments. And the question that we tried to answer that, okay, would glycerin increase the microbial biomass and then the activity while reducing that nitrate concentration? That was the first question. Second, whether and when that immobilized nitrogen in microbial biomass would be remineralized. Because if you think about it, we are binding in the microbial body, but if the plant, when they need that, if the soil or the microbes, they are not providing it to the plant, that means the plant will be starving. We do not want that. So that's another question that we try to answer. Third, would soil type affect the nitrogen mineralization or immobilization dynamics? So we'll see some data here. So what do you see here? Your x-axis, where we have the days after the glycerol application. Y-axis, we have the nitrate nitrogen concentration. And we have these 4 different rates of glycerol. One is the 0, or that's our control. Then we had 117 milligram per kilogram of carbon, then 468, and then 1872. Or in other words, if you want to remember this, low, medium, and then high is 3 rates of glycerol that we added. So here all the colors, so this is our control, or the 0. Then the lighter the colors are, that's how it's increasing the carbon content. And these 2 graphs, they are showing 2 different soil types. So what did we learn? First thing, glycerol lowered the nitrate nitrogen in both soils. So that means what was our hypothesis? It was accepted. So it worked actually. Second thing, nitrate is released around 20 to 38 days in 117 milligram per kilogram of carbon. You can see that from here, it started to release. But when we look at the higher rates of carbon, it did not release much, except here that some amount of nitrogen was released from the sandy type of soil. So last one, based on those 3 questions. So 2 questions we are answered. Third one, how the soils are going to respond. So 2 soils responded differently, even though they look, all these 2 figures look kind of the same way. Now the next thing is, depending on that nitrate, we saw that, okay, it decreased as the carbon content increased. So that means that carbon was actually storing all those nitrate. That means it immobilized, right? So we wanted to see the microbial biomass. So this is what it is showing you. These 2 types of soil in the first part, and we did collect the samples 3 times. One was at day 0, then at day 7, then at day 98, or around 100 days. And here we have these 4 different rates of our carbon, or the glycerol rates. So control 117, 468, and 1872. So the first thing that we learned, that the inorganic nitrogen increased over the 100-day incubation. Second thing, glycerol increased microbial biomass nitrogen in both soils. Again, our hypothesis got accepted. Third, higher rates of glycerin retaining most of the inorganic nitrogen. That's what we were expecting, or we would expect with that one. But 2 different soils, the responses were different. All right, so as I said previously, there are a lot of questions that we still do not know, but at least we know that it kind of improves our soil health because of the microbial biomass and the activity, because it increased that. We know that. It may improve the water quality. If that is the major question that you are trying to answer in your farm, it may work, but still we do not know how it is going to impact on yield. That's why I have that question mark. We do not know that. Another thing, when you are applying a lot of this carbon, there might be one condition which might happen, that, okay, maybe you are trying to improve your water quality, but at the same time, you might increase some nitrous oxide. We do not know that. So climate regulation, we do not know that whether it is going to work for that one. All right, so this is like in a nutshell, like all the examples that I thought would be important for soil health. How does that impact on nitrogen cycling as well as all other soil functions, and how does that improve the ecosystem services? Take home message, how we can improve our soil health. There are different management techniques. I did not cover all of them because I didn't work on all of those techniques. Some of those that I worked on, but there are a lot of other methods like no tillage, residue cover, which is very important. Strip tillage, there is another thing which is very important. Interseeded cover crops, which is also important. So I just got a grant from Minnesota Department of Agriculture, actually, and I'm collaborating with a farmer where we will be using some perennial grasses in between the corn rows, like instead of 60-inch, like we'll be using the 90-inch. All right, so we will see if we get some of those data. So hopefully next year or after 2 years, we will have those data. Maybe some of these we can answer after 2 years. So we still have a lot of room to improve. We cannot forget that one. We do not have a solid answer for that one. There are a lot of practices available, but we need more clarity. We need more research on that one. And I know a lot of the folks from Iowa State, from University of Minnesota and Midwest, all over the world, people are working on it. When you are looking to improve your soil organic matter, sometimes I get the call from the farmers like, okay, is it going to improve my yield? It may, but you need to think about that, okay, there is a potential that, okay, it will go up to a certain range. But you need to think about beyond that, that what are the other soil health benefits that you might get by increasing your soil organic matter. So look beyond that. Fourth, the most important thing, patience, which I know sometimes I get like, all right, I do not have enough patience while I'm doing it. And I don't blame the farmers if you are working on it. Of course, you are investing your time, your money, your blood, your sweat. You want the result right away. But I guess we cannot get that result right away. You need a lot of time. Last but not the least, policymakers, as I mentioned previously, I think they need to work. They need to provide proper incentives to the farmers so that they can implement all these different soil health promoting practices. All right? So that they can have enough farm profitability, whereas like they have positive impact on soil and then the environmental health. So with that, I would like to thank all my collaborators from Iowa State, from North Dakota State University, from USD ARS, and several undergrads, several graduate students that I worked with when I was at Iowa State. A lot of postdocs, lab manager, lab technicians, they helped me a lot. Otherwise, I didn't get all this data. And definitely the funding agency, without money, it was not possible to conduct all this research. So with that, thank you very much for the opportunity. So I'll take any questions if you have. I cannot. Do you want me to close this one? Oh, okay, I see. All right, before I start maybe answering these questions, is there anything from this room? Anyone has any question? No question from this room? All right, I will then start going through this chat. All right, first question. If you are limited by equipment or end markets, can cover crops help to diversify enough to make a meaningful difference in soil nitrogen, carbon? Is there enough growing time in Minnesota after harvest? Yeah, that's a great question. Yes, cover crops maybe get you some of those benefits. Like you all know that cover crops can improve the soil organic matter. At the same time, it can bind a lot of those nitrate. And if you are using some winter cover crops, when the next cash crop that you are growing, at that time, some of those nitrogen will be released. And that can be beneficial for your cash crop. The next one is, what would low, medium, high rates glycerin be per acre? I need to remember that one. I think it was 80 gallons per acre, the low rate, then 240 gallons per acre, and then so 80, 243 times. And I think the last one is 720 gallons per acre. But one thing, here is the thing. Don't take those numbers and start practicing right now. As I mentioned, that's our proof of concept. There are a lot of things that might not work at this point. I mean, we are getting a lot of great results. It's definitely promising. And Iowa State, now they have taken it to the field. They're working on it. So most probably a lot of results would be coming out of those data. And that might answer that, okay, what would be the optimum rate that we should apply if we are applying glycerol in our soils? All right. How many years were cover crops? Oh, sorry. There's another one. What causes PMN, PMC to decrease for a few years before increasing in the CRP example? Yeah, so so the PMN and the PMC, when we do that like lab-based incubation. All right, that means it's a very controlled condition. That's why we call it potentially mineralizable carbon or potentially mineralizable nitrogen. That means we are providing the potential condition. So in field, potential versus in the field, that might not work. So it's just some indicator which is showing you how much amount of your active carbon or the active nitrogen which is present in your soil, which is a good indicator if you want to see. Maybe another way, if I describe like the soil organic carbon which is present in your soil, there's like a slow changes that happen with that soil organic carbon. But the active carbon, they get highly affected by your management. So if you want to see the changes quickly, maybe those are the good indicators. So that's when you might start thinking about those. And some of the causes that might decrease, it could be that, okay, what are the plant inputs or like the rhizodeposits which is present in our soil, or all those plant materials like those restored grassland, how much amount of the rhizodeposits or the roots that is constantly exuding a lot of these amino acids, or a lot of other materials which is present in the soil, how they are feeding all the microbes within the soil, depending on that, that might actually cause that effect. All right. How many years were cover crops implemented? All right. So I'm not sure which project you are asking about. If it is my COPS project where I showed you the data at cover crop, like continuous corn with cover crops, I think that one started, I think 2008. And I started collecting the data in 2018. So you can think about like almost 10 years kind of. It's not that within 2 to 3 years, you will start seeing those results. It needs time. Again, patience. All right, what is the cost of glycerin? As I said, I cannot tell you right now all those things, but I believe it is going to be cheap. I hope I answered kind of all the questions, unless anybody has any other question here.
I think that's very, very good. Well, thank you.